My name is Kevin Roy. I am a research scientist here at the Stanford Genome Technology Center and in the Department of Genetics at Stanford University. Commercially, there are several new colony pickers that were built sort of in the same concept of using pins that would take colonies that were randomly grown across a plate and inoculate a multi-well plate filled with liquid. And all of these tools, from our standpoint, were limited in that we needed to have these pre-filled plates with liquid, which as you get to the scale of thousands and thousands of individual wells, you're dealing with large stacks of having to fill plates. And then ultimately we were going back to agar. And so we wanted a tool that would pick straight from an agar plate to an agar plate and not have to deal with the maintenance and cleaning of pins, which we knew had issues with accuracy and being bent, for example, when they were being cleaned. So the PIXL for us represented a transformative tool that would really simplify our workflow, allowing us to go directly from an agar plate to an agar plate at very high density. So minimizing the amount of hands-on manual labor that we need to do to put into the project with dealing with lots of plates. When we first started out using the PIXL, we had the standalone PIXL. And we had a technician that would work full-time and basically need to change out the plates manually. So in order for us to pick 10,000 colonies, we really needed a technician constantly checking on the system every 45 minutes to an hour to change out plates. And so we realized that we weren't tapping into the full capabilities or throughput of the system because there are 24 hours in a day, yet we were only using about 6 hours of those maybe for the actual picking process. So when the automated system first appeared, we jumped on it because we thought, here, this is what can get us to this scale of tens of thousands of variants. With the automated setup, we were able to get really ambitious and go for projects where we have several different strains, for example, up to 10,000 to 50,000 genetic variants between the strains, and we can begin to address what are the genetic variants that have a background dependent effect, what are the genetic variants that perhaps have interactions with other variants in one strain but not another. And so we were able to scale up the scope of these projects, and this is currently ongoing work in our lab. So here at the Stanford Genome Technology Center, we have many robots and many automated systems. So going into getting the PIXL automated, initially there are concerns about how much work this will involve on our end. At the time, we were one of the first labs to get the automated system. We weren't sure which robotic arm company should we partner with for this. And so we really relied on Singer and their technical support team to take care of a lot of the guesswork for us. We wanted a simple off-the-shelf solution where the automated system would be well integrated without too much work on our end. And our main focus was on high throughput colony picking. And we didn't need the robotic arm to be integrated with other systems. So we really depended on Singer to handle all of the details in terms of how the robotic arm software will integrate with the PIXL software. And it was really a seamless operation. Basically, even though we were one of the first to get the system developed, we really didn't have to do too much development on our end as a lab.
Have you ever been able to utilize the beer bottle opener in your PIXL?
Only after hours. There's one on the rotor too, so.
All right.
That's why it was placed near the edge of the table.
That's right. There you go. Okay.